Assalamu alaikum wa rahmatullah. In this lecture, we continue discussing the physiology of the gastrointestinal tract. And in this lecture, we start with the small intestine. The small intestine is the important part of the gastrointestinal tract. It is about 6.5 meter long. It is main part of the absorption. Absorption means movement of the nutrient and water electrolyte from the lumen of the intestine into the cell of the intestine, then from these cells to the blood. As we discussed in the previous lecture, there is no significant absorption occur in the mouth and esophagus, and also very small amount of absorption occur in the stomach. Some drug may be absorbed from the mucosa of the mouth like nitroglycerin, and also in the stomach some water and alcohol can be absorbed, while the main part of the absorption occur in the small intestine. The total quantity of the fluid entering into the intestine is about 9 liters. This 9 liters, 2 liters from the food and 7 liters from the secretion within the gastrointestinal tract. From these 9 liters, 7.5 liters are absorbed by the small intestine, and only 1 to 1.5 liters will pass to the large intestine. Small intestine food that entering the small intestine may be carbohydrate, protein, and lipid, and we discussed each part separately. There is a special modification in the small intestine for this action of which is the absorption. There is special fold inside the lumen of the small intestine. This fold increases the surface area, each fold about 1 centimeter long. In each fold there is a small projection called villi or villus. This villi is about 1 millimeter long. Each villus contain blood vessels and lymphatics that aid in the absorption of the nutrient from the inside the cell into the bloodstream. Also, each villus contain millions of the microvilli. These microvilli aid in the increasing the surface area, so increasing the absorptive surface. When discussing the absorption of each compound in the small intestine, we start with the carbohydrate. Carbohydrate digestion in the small intestine carbohydrate digestion in general start in the mouth with the action of the salivary amylase. There is no carbohydrate digestion in the stomach. When reaching to the small intestine, pancreatic amylase help in digestion of the carbohydrate. The carbohydrate form that absorbed by the small intestine is the monosaccharide. The monosaccharide is either glucose, fructose, or galactose. If the monosaccharide is present as monosaccharide in the nutrient, there is directly absorbed, no need for digest. If it is present in the disaccharide form, will be digested by the enzymes lactase, sucrase, or maltase into the monosaccharide and then will absorb on the brush border. While if it is present in the starch, then salivary and pancreatic amylase will digest these large compound into smaller and smaller until reach to monosaccharide and disaccharide that will absorb from the brush border. In this picture, in this slide, we see the mechanism of the transport of the glucose through the cell membrane inside the small intestine. It is by secondary active transport. In this, there is sodium. The sodium can pass freely from the high concentration inside the lumen to the low concentration intracellular. With the passage of this free passage of the sodium, it can pass with it glucose molecules. This process per se not need energy. But to continue this process to occur, sodium must continue to emptying from the intracellular. Emptying of the sodium from the intracellular to make the chemical gradient continue to acting. So the continuous acting of the chemical gradient of the sodium to enter the cell need the sodium to expel from the cell. The expel of the sodium from the cell need energy. So why is this type of transport called secondary active? Because for continuation of this type of the transport need energy from the other side of the cell. So the sodium will enter special protein transport in the cell membrane, and for each molecules inside in the of the glucose of the carbohydrate, there is special brush border molecules or special brush border transport protein. For the glucose and galactose pass through SGLT1, while fructose pass through GLUT5. All of them can pass through the basolateral surface of the epithelial cell from the GLUT2 to the blood vessels, and from the blood vessels we discuss when reaching to the liver. After finishing the carbohydrate digestion in the small intestine, we discuss the protein digestion in the small intestine. The protein digestion, there is no protein digestion in the mouth, and the protein digestion started in the stomach with the action of the pepsin. The gastric cell secrete inactive enzyme which is pepsinogen. This pepsinogen will activate it into pepsin with the action of the hydrochloric acid. The pepsin help in digest or breakdown of the protein into oligopeptide. This process continue in the small intestine with the action of the pancreatic enzyme. Many pancreatic enzymes help in digestion of the protein. This pancreatic enzyme also secreted as inactive form, which is trypsinogen, chymotrypsinogen, and procarbopeptidase. This will be activated by the epithelial cell intestinal mucosa, which secrete enteropeptidase. Enteropeptidase first will activate the trypsinogen into trypsin. Then the trypsin itself will activate more trypsinogen into trypsin, and also will activate other inactive enzyme which is the chymotrypsinogen and the procarboxypeptidase into its active form. And this will aid in complete digestion of the protein until reach the amino acid that will absorb through the epithelial cell into the blood cell. Now, when discuss the lipid digestion, the lipid is hydrophobic molecules, so it is poorly absorbed from the intestine. This hydrophobic need to be processed to be more hydrophilic or more easily absorbed. This processing firstly occur in the stomach with the aid of the acidity, also with the aid of the gastric lipase. But the gastric digestion of the lipid is also about constitute only 10 to 30% of the lipid digestion, while the remainder of the lipid digestion occur in the small intestine. In the small intestine, the bile acid help in the small intestine when the lipid entering from the gastric acid to the duodenum. The gallbladder secrete the bile. The bile will assist in defragment or make the lipid into small molecules. This process called emulsification. By this process, the bile have two surfaces. These two surfaces, one surface is hydrophilic and one surface is hydrophobic. The bile will combine to the lipid that make the hydrophilic surface is outside, and that fragment the lipid masses into small fragment. This will aid in the absorption of the lipid. These small molecules called micelles. The micelles reach to the brush border. The lipid inside the micelles after digestion with the pancreatic lipase and other pancreatic enzyme. The bile salt will return in the intestine, while the lipid molecule with the small amino with small glyceride are absorbed in the intestine, and the bile salt will retain in the intestine, stay in the intestine, and then will absorb in the terminal ileum by circulation called enterohepatic circulation. After discussing the small intestine, now we're discussing the large intestine. From its name, it is larger and thicker than the small intestine. It is about 1 to 1.5 meters. It is encircle the small intestine, occur around the small intestine. It consists of cecum, colon, and the colon consists of ascending colon, transverse colon, descending colon, and sigmoid colon, rectum, and then the feces passes through the anus. Also, there is a special modification of the function to large intestine to do its function, which is absorption of the small amount of water and vitamin and other ions in the large intestine. The large intestine have no projection like in the small intestine. Also only have small mucosa, which lined with the mucosa, which with the crypts of lubricants, and also contain gland that secrete mucus. This goblet cell gland secreting mucus called goblet cell. This mucus is very important in protecting the mucosa of the large intestine from the masses of the feces, which is more hard, more fiber content, so may affect or may injure the mucosa of the large intestine. Also, inside the large intestine there is a large number of bacteria, which is normal flora. It is bacteria, but it's not pathogenic bacteria. It is normal flora. This normal flora have very important function in the digestion and secretion of other nutrition, and can also it is important in production of the vitamins. This is the mucosa of the large intestine. There is no villi, many goblet cell, which is mucus secreting cell, and many surface absorptive cell, which is absorb water, and this is the crypt of the lubricant. After the food are processed in the small intestine, in the large intestine, many water and fluid and ions are absorbed in the large intestine till reaching the rectum. In the rectum, the defecation of the feces will occur. In the rectum and the anus, there is two sphincter: internal sphincter, which is involuntary, and external sphincter, which is voluntary. When the food particle or the feces reaching to the rectum, there is a special receptor will activate many reflexes that will relax the internal sphincter. If the defecation is not desired, so the external sphincter can close the anus and prevent defecation, while when the defecation is desired, so the defecation can occur, and this processes need many steps, which is relaxation of the external sphincter, contraction of the muscle, abdominal muscle will aid in the defecation, and also relaxation of the pelvic muscle. It's important to say that is absorption of the molecules and substance within the large intestine. The large intestine reach the food particle reach to the large intestine, which is concentrated with low water. So absorption of these substances need active transport for aiding and many mechanisms for aiding of the absorption of the water and the ion, and this will discussed in the lecture. After discussing the small intestine and large intestine, now we will discuss the gallbladder. Gallbladder is a spear-shaped organ occur in the abdominal cavity. It is for storage of the bile. The bile is secreted from the liver. The bile not secreted in the gallbladder. The gallbladder only the storage for the bile. While the secretion of the bile it is from the hepatocyte. Bile is an aqueous alkaline greenish to yellow liquid whose main function for emulsification of the fat in the small intestine, and also the bile can eliminate some substance from the liver. The body can secrete about 1/4 to 1 liter per day. Bile consists of bile acid, cholesterol, phospholipid, and bile pigment. Also electrolyte and water. Bile is secreted continuously by the liver, but it is stored by the gallbladder, and gallbladder contain contractile muscle that can push the bile through special duct into the small intestine via ampulla of water, which share the same entrance of the pancreatic duct. This is the main pancreatic duct, and this is the gallbladder. The gallbladder receive its bile from the right and left, and then hepatic duct stored in the gallbladder. Then when the contraction of the meal by many reflexes, expel of the bile into the common bile duct, which meet the pancreatic duct, and emptying in the duodenum. When there is ampulla of water. Stimulation of the contraction of the gallbladder and the sphincter of body by many reflexes. As we say in the previously with the absorption of the fat, bile aid in the absorption of the fat by the emulsification and making micelles, which is small droplet. The fatty acid will be absorbed in the small intestine while the bile will retain. Then the bile will continue to pass till reaching the terminal ileum, where it is absorbed and then return to the liver through portal vein. This process called enterohepatic circulation. This is the liver secrete the bile, and the bile by common hepatic duct will stored in the gallbladder, then secreted with the duct of the pancreatic to the duodenum, aid in the absorption and digestion and absorption of the fat. Fat will be absorbed while the bile will retain in the small intestine till reaching to the ileum, and then in the ileum will be absorbed to the portal vein, and then will pass to the return to the liver. Now we discussing the pancreas. Pancreas is important abdominal organ. It's have both exocrine and endocrine, and as we say in the previous lecture, exocrine mean gland with duct, while endocrine mean gland with no duct. So the pancreas have two functions. The main function is the exocrine, with secretion of the secretion of the digestive enzyme, and by weight about 85% of the pancreas is the exocrine gland. The pancreatic gland have many acinas and duct. The pancreas secreting digestive enzyme and also secrete water and bicarbonate. The bicarbonate is also important in digestion and also in neutralization of the acidity of the stomach and the small intestine. Secrete many enzymes like protease, pancreatic lipase, amylase, and also bicarbonate. And also many factors help in triggering the pancreatic to secrete its secretion, which is maybe vagal innervation, secretin release, and cholecystokinin. Other important organ in the body is the liver. The liver is the second largest organ, located in the right upper quadrant of the abdomen. It is largest visceral structure. It is the largest gland in the human body. It has many functions. As discussed in the previous lecture, it is store and processing the nutrition coming from the small intestine. Also help in detoxification of the virus, metabolic drugs, and other substances. Also help in the synthesis of the protein and produce biochemical necessary for the digestion and growth. The liver receives its blood from two sources. First source is hepatic artery for supply oxygenated blood for the liver and constitute about 20% of the blood supply of the liver. Other blood supply is from the portal vein. Portal vein as its vein, so it is deoxygenated blood, but it supplies the liver with a nutrient that's absorbed in the small intestine for more processing in the liver. Many functions of the liver, and each will discussed in the lecture. Important to say, there is in the blood supply of the liver, there is no capillary as discussed in the previous lecture. There is from the hepatic artery, from the portal vein, directly path this blood path the hepatocyte till reaching to the central vein, and then after the processing of the nutrient and content of the blood, the blood pass through the hepatic vein. Carbohydrates, fat, and protein are metabolized in the liver, and these molecules absorbed from the small intestine can be stored or used for the production of the other compound important for the function of the body. And different mechanisms aiding in the metabolism of this compound occur in the liver. Also, multiple vitamins, especially lipid-soluble vitamin, which is A, D, E, K, and also vitamin B12, are metabolized in the liver. Thank you for your listening.